All right, welcome to 3.2 part 2. So this is going to look at the application statements that you wanted to look over, which is 1, 2, 3, and 4. All right, so A1 is really looking at the Cairns technique for measuring the length of DNA molecules via autoradiography. Okay, so basically John Cairns produced an image of the DNA molecules from E. coli. So what he did is he grew E. coli in a thymine solution that had a radioactive isotope of hydrogen. Okay, so you have these radioactive thymines. And what that meant was that when the E. coli was growing, it would incorporate that radioactive thymine as it replicated its DNA, right? So what that caused was it caused the DNA to be labeled because they could see it using a particular technique that would make the radioactive part visible. So the E. coli cells were then broken open by enzymes to release the cell contents, and then the cell contents were applied to a photographic emulsion and placed in the dark for 2 months. What happened was that the radioactive isotopes reacted with the emulsion, very much like the way that light does when we do darkroom photography stuff. And the dark areas of the photographic emulsion indicated the presence of DNA. Yeah. So the images showed that E. coli possessed a single circular chromosome. They were able to measure it and found that it was 1,100 micrometers long. And the Cairns images provided some evidence to support some of the semi-conservative replication theory statements. Okay, so that's really all it is just understanding that this is how the technique works. It's used through adding a radioactive isotope for the nucleic acids, so that as that nucleic acid gets incorporated, we can then use a photographic technique to actually identify the length of DNA. Okay, A2 was about comparison of genome size in T2 phage, E. coli, Drosophila, Homo sapiens, and Paris japonica. So this is just looking at different genome sizes to reiterate the fact that genome size does not necessarily correlate to complexity, but just to get some ideas of what these numbers actually would be. So humans have 3.2 billion base pairs. Remember that genome size is total number of DNA base pairs in one copy of a haploid genome. Okay. Now we can look at some other organisms. We have the canopy plant, or Paris japonica. We have the T2 phage, which is the orange part here. So you have one there, and one there, and one there. And it's a virus that attacks E. coli. E. coli is this green and white blurb thing here. This is Drosophila, which is a fruit fly. Okay, and with these guys, the Paris japonica, the canopy plant, has 150 billion base pairs. To remind you, humans have 3.2 billion base pairs. All right, the fruit fly has 130 million base pairs. The E. coli has 4.6 million base pairs, and the T2 phage, right, the virus, has 164,000 base pairs. So why is this important? Well, one of them is to realize just how many that actually is, right? Billions that's a really big number. And even 164,000 base pairs, there's a lot of information in that still, okay? So just to get an idea of the differences in genome size and how big that is, you should certainly know what the human genome size is. That's helpful. Now, A3 is chromosome number. So we just looked at, sorry, there's some like PowerPoint issues here where the number is below that circle. We just looked at base pair numbers, so genome size. We're going to look right now at chromosome number. Okay, so we said that humans have 3.2 billion base pairs. What that ends up equaling to for humans is 46 chromosomes. Okay, and then in rice and roundworms and dogs and chimpanzees, they're going to have different numbers. Yeah, and so rice has 24 chromosomes, and roundworms have 2, and dogs have 78, and chimpanzees have 48. So this is important because, again, looking at these numbers and doing a comparison and how they don't really tell us the whole picture is important, right? But the fact that 2 chromosomes can tell us all the things that we need to give us all the information for this little roundworm, that's kind of important. I will go back and note that chromosome number is diploid chromosome, right? Okay. A4 is karyorgrams. See, I told you we'd come back to this. So this is really about looking at karyorgrams to deduce sex and any possible diseases. The biggest one that IB wants you to know is Down syndrome. So can you use a karyogram to determine sex and whether a person has Down syndrome? Well, if you look here, we already did this in class, so just a quick review of the fact that you can identify Down syndrome by looking at chromosome 21. Down syndrome is caused by trisomy 21, which is where you have a 3rd chromosome, where you should just have 2. There are many other genetic diseases caused by trisomy, but you don't need to necessarily know those ones. In addition, can you identify sex? Yes, you look here, it's going to be the last one in the karyogram. This has an X and a Y, so this is a male with Down syndrome. If there were 2 X's and nothing in the Y category, that would be a female. Okay, and that is it for the application statements.